Hi everyone. So today I'm going to talk about ICH guideline Q1R2, which is stability testing of new drug substance and products. Okay, so if you would like, I can explain in Hindi too, or I can use both languages. So let me know. Okay, so what is this guideline? This guideline is developed by ICH, um, and it provides comprehensive recommendation on how to design and conduct stability studies to ensure safety, quality, and efficacy of new drug substances and their corresponding drug products over time. So let me go on the different sections. So, what is the primary goal of the guideline? It is to establish a retest period for drug substances and a shelf life for drug products by evaluating their stability under various environmental conditions, including temperature, humidity, and light exposure. The guideline harmonizes the requirements across major regulatory regions, including US, EU, and Japan. Okay. So, what is the scope of this guideline? Okay, if you want, I can explain a little bit in Hindi. So, जब हम कोई भी ड्रग प्रोडक्ट यानी मेडिसिन लेते हैं तो हम देखते हैं कि उस पर एक एक्सपायरी डेट होता है. तो वो एक्सपायरी डेट कहाँ से आता है? तो उसके लिए उस ड्रग प्रोडक्ट को, उस मेडिसिन को स्टेबिलिटी टेस्टिंग पे रखा जाता है और उस उसके रिज़ल्ट के अकॉर्डिंगली उस पे जो एक्सपायरी डेट होती है वो दी जाती है. ओके, सो व्हाट इज़ द स्कोप ऑफ दिस गाइडलाइन? It provides instruction on what information companies need to include when applying to get approval for new drugs and their related products. Okay, but this guideline doesn't cover application for generic drugs, um, or changes to existing drugs, like if there's a variation in manufacturing process or something else. So that is not included in this guideline, and it doesn't include the approvals for clinical trials. Now, if you want to know more about what is generic drug or new drug or clinical trials, I can explain it, but in some other videos. Let me know if you want to know more about clinical trials and the difference between uh, new drugs or generic new drugs and generic drugs, which is NDA and ANDA. Also, this guideline does not go into detail about how to sample and test specific types of drug products in their container. Okay, it doesn't talk about more like a lot of details about how to sample and test. It just gives a guideline of the like what are the storage conditions required when you're going to do the testing, all those things, and how, like what should be the container and closure system for the drug substance. So if the companies are working on new dosage form, okay, like they are like they now they have a tablet and they are changing it into a liquid form, or biotech, a biological product such as vaccines or insulin. So they should check other ICH guideline, which is Q1C for new dosage form and Q5C for biotech and biological products. Now, general principle. The main goal of stability testing is to check how a drug's quality changes over time when exposed to different environmental factors like temperature, humidity, and light. Okay. So how long a drug substance can be, and by this testing, it will help to determine how long a drug substance can be stored before it needs to be tested again. How long a drug product remains safe and effective for use, which is expiry date, and what is the best storage condition for the product. The testing conditions recommended in this guideline are based on the typical climatic condition in Europe, Japan, and the United States. Now we know the world is divided into four climatic zones, one, two, three, four, based on temperature and humidity. But this guideline mainly focuses on zone 1 and 2, which represent cooler and temperate climate. An important agreement is that let me if a drug passes stability test in one of these three regions, the same data is generally accepted in other two regions as long as the data follows the guidelines requirement and the drug labeling meets the regional regulation. Okay. Now, what is stress testing? So, how do we do stability testing? So, we put stress on those drug substance in this drug product, and see whether they are changing physically, chemically. Okay, and then we decide like, okay, so this is having effect on the drug substance. So, stress testing is a process where a drug substance is exposed to extreme conditions to check how it breaks down and what kind of degradation products are formed. This helps to identify degradation pathways, like understand how the drug breaks down, to evaluate stability, check how stable the drug is under harsh condition, and to validate analytical methods to confirm if the testing methods can accurately detect degradation products. Okay, so that's called stress testing. Now, how it's done? Stress testing is usually done on one batch of the drug. Okay, so what are the condition? Higher temperature, like 50 degrees, 60 degrees. High humidity, okay, which is 75% RH. And then oxidation, the drug substance is exposed to oxygen. Photolysis, it is exposed to light. And also we check here how the drug reacts in different pH solution, which is acidic, neutral, or alkaline. And for photostability testing, that is light exposure, we have a different other guideline, ICH-Q1B. Okay, so if stress testing shows that certain degradation products are not formed during normal storage, further testing for those products may not be necessary. Okay, the result of these tests are essential for regulatory submission to prove that the drug remains safe and effective throughout its shelf life. Now, this next section is selection of batches. How do we select batches for stability testing? So, when a pharmaceutical company wants to prove that a drug remains stable over time, they conduct formal stability studies. For this, they need to provide data from at least 3 primary batches of the drug substance. So, what is primary batches? So, these are main representative batches chosen for stability testing. The batches should be manufactured on a scale large enough to closely simulate the actual production process. Okay, while it's not full scale production, it is a significant step up from laboratory experiment. Okay, so what first is laboratory? In laboratory, they do like many small scale production. Then they go to primary batches, and that primary batches data, stability data is sent to FDA. And once they get approval, then they start making production batches. So these primary batches, they should be exactly same as the final batches. The batches must be made using the same chemical process and production method that will be used for future commercial production. Okay, so what is the goal here? The goal here is to ensure that the stability data accurately reflects how the drug will perform when it's made on a large scale. Okay, companies may also submit additional supporting data to provide further evidence of the drug's stability if necessary. Now, what is container closure system? So if you see any drug substance or drug product, So they are packed in some type of container or aluminum foil or blister packing. So those are called container. The packaging thing is called container closure system. So the stability studies need to be done using the same type of container and packaging that will be used when the drug is actually stored, shipped, and sold. If using the exact packaging isn't possible for testing, they can use a similar one that mimics the actual storage condition. This will ensure the results reflect how the drug will perform in real world scenarios. Okay, container closure system. Now, specifications. So, what are specifications? Like, how do we decide whether the drug is uh, undergoing degradation or drug is stable? So they do some tests, and then they have some acceptance criteria for each test. Like, suppose initially the drug is 100% pure, we say that we get assay as 100%, like purity test, which is assay. But as the time goes, it's possible that due to light or moisture, um, like temperature, it's possible that the drug will go degradation. Okay, and then it is not 100% pure now. Now it will have like as say as 98%. But what about the 2%? Maybe it's there's an impurity formation. Then 2% is impurity. So now specification is it tells you like the assay should be initially should be 100%, not more than not less than whatever 90%, 95, 99%, like that. So suppose if the sample is failing in that acceptance criteria, so that means the product is not stable. So that is called specification, that it should be um, within that acceptance criteria. So these specifications are a set of tests, methods, and acceptance criteria used to ensure the quality of a drug substance. Okay, the guideline ICH Q6A and Q6B cover these specifications. So what should be the specification? Like all those things are mentioned in the different like other guideline, which is ICH Q6A. While ICH Q3A specifically discuss the limit for degradation products. Okay. So, when conducting stability studies, companies should test the properties of the drug that might change over time and affect its quality, safety, or effectiveness. These tests can include checking the drug's physical appearance, chemical composition, biological activity, and microbiological properties. For example, physical appearance, like sometimes color might change, or the tablet becomes like soft, or if there's some ointment or cream, you see like the cream is not uniform, you see a face separation. So that's all physical appearance. But at the same time, maybe it's degradation is going on there. So that is a chemical composition, some biological activities going on. So all these things will be checked during the stability studies to ensure. And then how they are going to test these physical properties, they have to use some methods like right, like HPLC, GC, pH meter, Carl Fisher, like these are the instruments to check water content. And then we have to use a method. You cannot just keep the sample under instrument and just get the result. You have to do some preparation to get the result, like dissolve in something and then put in HPLC, select a wavelength, all those things. So that method should be validated. Okay, you cannot just use any method. And validation means you have to make sure the method will be able to find detect the impurity even at the 36 months. Uh, so the need for and The need for repeating tests like replication will depend on how well the methods perform during validation. Okay, so this was specification. Then we'll talk about testing frequency. So there are three conditions. Okay, first is the long-term stability study. Long-term means like normal, whatever is in zone like 25 degrees, 25 degrees temperature and 60% humidity. And then there's an accelerated stability study. Okay, first for long-term. So there are three conditions. Long-term stability studies, the testing frequency should be enough to understand how the drug substance remains stable over time. For substance with a proposed retest period of at least 12 months, the testing schedule should be every 3 months during the first year, every 6 months in the second year, annually after that for the duration of the retest period. So suppose if there's a drug substance and the retesting period is, for example, 4 years. Okay, so how we are going to test the samples, like what should be the frequency. uh, so initially it will be 0, 3, 6, 9, 12, like after every 3 months. So once it is 12 months, then it will go after every 6 months. So after 12 months it will be 18 months and then 24 months. Then second year is done, and then after second year annually. So after 24 months it will be 36 months. So 3 years is done, and then it will be tested on fourth year. So 36 plus 12 whatever. So that should be the testing frequency. This is for long term. For accelerated stability studies where conditions are more stressful to speed up aging or degradation, okay, the guidelines suggest at least 3 test points: 0, 3 and 6 months over a 6-month study. If there is a possibility that the result will show significant changes, it is recommended to test more frequently, either by adding extra samples at the final test point or adding a fourth test point. Okay, so if the accelerated storage condition shows that the drug is changing significantly, then intermediate storage condition, okay, which are less harsh than the accelerated condition, should be tested. For this intermediate study, the guideline recommends testing at 4 time points at the start, 0 months, and then 6, 9 and 12. Okay, this helps assess the stability of the drug when stored under these less extreme conditions. Then we go to storage condition. What should be the storage condition? I think, okay, a drug substance should be tested under storage condition that checks its thermal stability and, if necessary, its sensitivity to moisture. The strong condition and study duration should be enough to cover how the drug will be stored, shipped and used after that. Okay, so that's what I already mentioned: long-term stability, at least 12 months, using at least 3 primary batches, okay, of the drug when submitting the application. This testing should continue for as long as needed to cover the proposed retest period. Any additional data collected during this time should be provided to authorities if requested. Okay. Now, accelerated condition, again, it is a harsher condition, and like they have, and if necessary, intermediate storage condition can help evaluate the effects of short-term storage changes like those that may happen during shipping. Okay, the guideline for long-term accelerated intermediate storage conditions are explained in more details. Okay, if the drug in the following section, if the drug substance doesn't fall under a specific section, then general rules apply. Okay, alternative storage conditions are allowed if they are justified. So if you see 2.1.71, its long term is 25 degree, temperature is 25 and humidity is 60%. for 12 months, then intermediate is 30 and 65 for 6 months, accelerated 40 and 75 for 6 months. And it is up to the applicant to decide the long term. If they are doing long term at 30, then they don't have to do intermediate. Okay. Long-term studies are conducted at 25. Okay, if significant change occurs during 6 months of accelerated testing, which means the drug fails to meet its specification, then additional testing and the intermediate storage condition must be performed. Intermediate storage condition testing should include all the necessary tests unless there is a valid reason not to. The initial application should include at least 6 months worth of data from a 12-month study conducted at the intermediate storage condition. And what is significant change? Means the drug fails to meet the specification it should meet. Now this is a table for drug substance that are stored in a refrigerator. For that the long-term temperature is 5 degrees and the accelerated is 25. Okay. So if there's a significant change between 3 and 6 months during accelerated testing, the retest period should be determined based on the actual data from the long-term storage condition. If the significant changes occur within the first 3 months of accelerated testing, a discussion is needed to explain the possible effect of short-term deviation from the recommended storage conditions like during shipping or handling. If needed, further testing can be done on a single batch for a shorter period, which is less than 3 months, with more frequent testing than usual to support the discussion. If a significant change happens in the first 3 months, it's not necessary to continue testing through the full 6 months of accelerated testing. Okay, because now you know that your product won't be stable. If it is failing in 3 months, there's no use of doing continue for 6 months. So this table is drug substance intended for storage in a freezer, which is -20. Okay, for drug substance stored in freezer, the retest period should be based on the real-time data from long-term storage at the actual storage temperature. In the absence of accelerated storage condition for drug substance stored in a freezer, testing a single batch at elevated temperature like 5 degrees or 25 degrees should be done for a suitable time period. This is to check how the substance react to short-term temperature changes, such as what might occur during shipping or handling. Okay, and for the for drug substance stored below -20, they should be treated individually, and the decision will depend on the specific substance and its requirement. What is stability commitment? So when long-term stability data does not cover the proposed retest period at the time of approval, so suppose a company is submitting its application, it's not and it not complete the whole period up to the retesting period, like they have 24 months retesting period, but they have submitted only data up to a year, then they should make a commitment that they will continue the stability studies after approval to confirm the retest period. So the submission includes long-term stability data for 3 production batches that cover the proposed retest period. A post-approval commitment is not needed. However, if not, so what is the 3 production batches means? 3 production batches means commercial batch. Like once the drug is approved, the company will start manufacturing product for people, right? So that is called commercial batch or production batches. Then again, they will have to keep 3 commercial batches on stability, and they have to submit that data to FDA, inform FDA that okay, it is passing or not passing. So they have to, so when they are submitting their application initially, they have to make that commitment that they are going to do those three production batches. So if they are already completed, they don't have to do this commitment. However, if not, one of the following commitment must be made. If there is a data from at least three production batches, a commitment should be made to continue the study for the full proposed retest period. Suppose they started doing the commercial batches stability. Okay, they are not released in the market, but they have kept those batches on stability, but it's not complete. The stability study is not complete. So they will make a commitment that we are going to continue the stability study up to the retest period. Okay. If there's a data from fewer than three production batches, suppose they have only two production batches data, so they are going to do the commitment that they are going to complete all three batches. Okay, and they'll do the stability study, all three batches, and the long term studies. If there is no data on production batches, okay, a commitment should be made to place the first three production batches on long term stability study covering the full proposed retest period. Okay, and the stability protocol used for these long term studies should be same as that for the primary batches unless there's a valid scientific reason to change it. So when they submitted primary batches, they submit stability protocol with that. And now if they want to do the production batches, the protocol should be same as that primary batches. Okay, if they're doing any changes, it should be justified, and they should inform FDA about it. What is the reason and why they have submitted, why they've done the changes. Now, evaluation. Purpose of stability study is okay. The goal is to establish a retest period for the drug substance based on testing at least three batches and evaluating various stability attributes, which is physical, chemical, biological, microbiological. The stability data helps determine how long future batches of the drug will remain within the accepted specification. Okay, so if there's a minimal degradation and little variability between batches, okay, it may be clear that the requested retest period will be granted. In such cases, formal statistical analysis may not be necessary, though justification for this omission should be provided. Now, what is statistical analysis? For quantitative attributes, example drug content, expected to change over time, the 95% one sided confidence limit for the mean curve should be used to determine when the drug will no longer meet acceptance criteria. If variability between batches is small, it may be possible to combine the data into a single estimate. This requires testing the slope of regression lines and intercepts for individual batches using statistical methods, example p-value. If data cannot be combined, the retest period should be based on minimum time a batch can stay within specification. Okay, and then when they are using extrapolation, okay, suppose their stability study is not complete and they are doing this method to know what will be the shelf life or retest period, so they can do that method, uh, uh, but they can use that method only if justified. Okay, and the justification should consider uh, the degradation mechanism, accelerated testing results, how well the data fits the mathematical model, batch size, and other supporting data. Okay, and it says, however, this extrapolation assumes the same degradation pattern will continue. The evaluation should cover more than just the assay. So it should account for the degradation product and other relevant impurity, not just purity like impurity, um, pH, water content, uh, like all those things. It should consider everything, not just the assay. Okay, so now statements labeling. Storage statement. Uh, so the labeling of the drug substance should include a storage statement that complies with the relevant national or regional regulation. The statement should be based on the stability data collected during testing. So suppose the stability data shows that um, your drug substance is not stable, um, like in light, so in storage condition, then you will have to change the packaging. You have to mention in storage condition. You must have seen a lot of drug like drug products packed in amber colored bottle or amber colored for blister packing. That means they are not stable in light. So they should mention in that storage statement. Okay. Um, the label should include specific instruction for the drug substance, particularly for those that cannot tolerate freezing, like do not freeze. You must have seen. These instructions should be clear to ensure proper storage condition. And they should not use general terms like ambient condition or room temperature in the storage instruction, because these terms can be too broad and may not provide enough clarity on how to properly store the drug substance. Then the retest period, how long the drug can be used before it needs to be tested again, should be derived from the stability data. Okay. If appropriate, a retest data should be indicated on the container label, helping to ensure the drug substance is used within its effective period. So, this was for the stability guideline for drug substance. Okay, so in maybe in part 2 I'll be covering the guideline for drug products, so make sure to stay tuned. um, Thanks for watching, and I'll see you in the next one. Thank you.